Hello! In today's video, we shall learn in detail about Pteris fern. By the end of this video, you will have a clear understanding about its morphology, anatomy of root, rhizome, leaflet, petiole, reproduction, development of sporangium, gametophyte, Antheridium, Archegonia, embryo, and alternation of generations. Let us begin by studying the morphology of Pteris, a genus of ferns, commonly known as brake ferns belonging to the division Pteridophyta. They are a group of terrestrial, highly advanced cryptogams inhabiting shady and moist places. The main sporophytic plant body is differentiated into root, rhizomatous stem, and leaves. The primary root is ephemeral, short-lived, and is replaced by a large number of adventitious or fibrous roots which grow profusely from the rhizome. The underground stem is branched, perennial, rhizomatous, and surrounded by brown scales called ramenta. The underground stems are generally known as rhizome. The rhizome may be creeping as in Pteris grandiflora, or it may be branched, short, and erect as seen in Pteris critica. Leaves are usually pinnately compound, macrophyllous, arising in acropetal pattern on the rhizome and succinate. The leaves of fronds show open furcate venation in which the veins branch out in a forked manner and do not form a network. The petiole in Pteris ferns are covered with scales or ramenta at the base. It has an adaxial groove with vascular bundle and dorsolateral ventilation areas specialized for aeration. Reproduction in Pteris is through vegetative and asexual methods. Pteris exhibits alternation of generation in its life cycle. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn, and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Let us study the transverse section of Pteris root. The Pteris root shows the following layers. The epiblemma or the piliferous layer is made up of a single layer of cells. Beneath the epiblemma is the cortex. The outer cortex is made up of thin-walled parenchymatous cells, while the inner cortex is made up of thick-walled sclerenchymatous cells. Inner to the cortex is the stele, which has a central core of xylem surrounded by phloem. In Pteris, the xylem in the root is organized in a diarch and exarch manner. Next is transverse section of Pteris rhizome. The TS of Pteris rhizome shows epidermis, cortex, and stele. The epidermis is single-layered, covered by thick cuticle. The cortex consists of a few layered thick sclerenchymatous hypodermis and a broad parenchymatous cortex. The stele has xylem in the center and is surrounded by phloem. The stele is bounded by endodermis and pericycle. The endodermis shows the presence of Casparian strips. Inner to the endodermis are one or two layers of cells forming the pericycle. Stellar organization is diversified, It may be solenostylic or dictyostelic. The stele is made up of a number of meristeles. Each meristele has a plate-like mesarc xylem surrounded by phloem. Next is the anatomy of Pteris leaflet. Leaflet consists of epidermis, mesophyll, and vascular bundle. Both the upper and lower epidermis are single-layered, and the stomata are restricted to the lower epidermis. The mesophyll contains lower spongy layer and upper palisade or homogeneous layer. Midrib shows the presence of concentric amphicribal vascular bundle in which the xylem is surrounded by the phloem. The vascular bundle is surrounded by a single layer of pericycle and endodermis. Vascular strands are embedded in the mesophyll. Next is the anatomy of Pteris petiole. The vascular bundle in petiole of Pteris is typically C-shaped, U-shaped, or V-shaped. It is referred to as a leaf trace. Xylem consists of protoxylem and metaxylem surrounded by phloem. Pericycle and endodermis form the bundle sheath. In the rachis, if the leaf is unipinnate, the traces form strands, and if the leaf is bipinnate, it forms secondary and tertiary traces. The traces seen in rachis are flat, U-shaped, and marginal in origin. Now, let us understand reproduction in Pteris. Pteris can reproduce by vegetative means. When death and decay of older sections of rhizome takes place, the young parts of rhizome separate and give rise to new individuals. In Pteris, asexual reproduction is through spores found on the undersurface of the leaf or sporophyll. The sporangia are grouped together in structures called sori. The sorus of Pteris is Cenosorus. The sori are marginal, linear, and continuous, protected by the reflexed margin false indusium of the pinna. Pteris produce only one type of spore, making them homosporous. Many sterile hairs are present in between the sporangia. Next, let us study about the development of sporangium in Pteris. A single sporangial initial divides transversely, forming an upper cell and a lower cell. The upper cell, by oblique division, gives rise to the apical cell and lower portion, which later form the stalk of the sporangium. The apical cell divides to form an outer jacket initial and an inner tetrahedral archisporial cell. The archisporial cell on division gives rise to the tapetal initial and the primary sporogenous cell. The tapetal initial forms a double-layered tapetum. The tapetum nourishes the spores. The primary sporogenous cell forms the spore mother cell, which undergoes meiosis to form haploid spores. A mature sporangium in Pteris has a long stalk that terminates in a capsule. The capsule wall is thin but has a thickened cutinized band of ring called the annulus, which encircles the sporangia. Radially arranged thin-walled portion of the annulus is the stomium. The spores are homosporous, triangular, thick-walled, with sculptured outer exine and inner intine. When the spores mature, the sporangium splits transversely along the stromium due to shrinkage of cells of annulus. The spores are liberated and dispersed by wind. Next is the development of the gametophyte. The spore germinates by rupturing the exine. The inner contents form the prothallus cell and a rhizoidal cell. The prothallus that develops from the prothallus cell is a haploid, small, green, heart-shaped structure. It bears both reproductive organs, namely Antheridia and Archegonia. Prothallus of Pteris is monoecious and exhibits protandry. Prothallus is short-lived, independent, dorsiventral and flat. Rhizoids develop on the ventral surface of the prothallus. Antheridia are present on the ventral surface of the prothallus and originate from the antheridial initial. The antheridial initial divides into lower cell and upper cell. The lower cell forms the first ring cell and an upper cell divides periclinally to form a dome-shaped cell and a primary androgonial cell. At this stage, the young antheridium consists of a dome-shaped cell, a primary androgonial cell, and a ring cell. The dome-shaped cell undergoes transverse division forming cover cell and a second ring cell. Both divide to form a single layered jacket around the antheridium. The primary androgonial cell divides to form several androcytes which give rise to the anthrozoids. A mature antheridium has 2 ring cells, a jacket, and a mass of coiled ciliated anthrozoids. The Archegonium is a flower-shaped structure on the prothallus. The swollen basal portion is known as the venter and the slender tube-like upper portion as the neck. The Archegonium arises from the archegonial initial. The archegonial initial divides to form an outer primary neck cell and an inner cell. The outer primary neck cell divides anticlinally to form neck cells. The inner cell further divides to form an upper central cell and a lower basal cell. The central cell gives rise to the neck canal cell and the venter cell. The neck is short and curved. The egg develops in the lower portion of the archegonium within the venter. Let us study about fertilization in Pteris. At maturity, the antheridium absorbs water and swells. A pressure is created within the antheridium due to which the cover cells split and the anthrozoids are released in water. In the archegonium, the ventral canal cell, the neck canal cell, and the neck cells disintegrate. An open passage is formed for the entry of anthrozoid. Mucilage is produced and one of the anthrozoid enters and fuses with the egg to form the zygote. Next is the development of the new sporophyte or the embryo. The zygote first divides vertically, followed by a transverse division, resulting in the formation of a quadrant. Repeated divisions form 32-celled embryo. At this stage, a proper differentiation of the embryonal organs take place. Stem apex and foot arise from the hypobasal cells, while the cotyledons and root arise from the epibasal cells. Venter of Archegonium forms a protective layer around the developing embryo known as the calyptra. The embryo soon develops into a young sporophyte. The prothallus decays and the young sporophyte grows into a fern plant. Finally, we shall learn about the alternation of generations in Pteris. Pteris exhibits an alternation of generations to complete its life cycle. It has two distinct phases, a diploid sporophyte and a haploid gametophyte. The sporophyte is dominant, independent, and reproduces asexually by spores. These spores develop into the gametophyte or the prothallus. The prothallus reproduces sexually by producing male and female gametes. Fertilization of the egg by the sperm results in a diploid zygote, which gives rise to a new sporophyte or the fern plant. So today, we have learnt in detail about the morphology, anatomy, and alternation of generations of Pteris fern. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notifications of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.